Today we are going to discuss future of man, future of Homo sapiens, future life. Life has been evolving. Homo sapiens also must be evolving. But we don't know exactly how Homo sapiens are evolving and what they will evolve into. But there is a proposal made by Yuval Noah Harari that Homo sapiens may evolve into Homo Deus. The title of his book is Homo Deus, A Brief History of Tomorrow. So Homo sapiens evolving into Homo Deus. He called them like that because he thinks genetics is going to contribute to evolution of man with very desirable features. So using godlike technologies. Something like designer babies. I want my child to be more intelligent. I want my child to be taller or with talent for music or some other athletic ability. So he thinks that genetics may be moving in the direction of shaping human evolution, Homo sapiens becoming Homo Deus through genetics. So we are going to examine that. What is the most recent development in this aspect? See, Harari thinks that evolution may take place through what is called germline editing. Germline editing. That is, editing zygote or embryo. And embryo editing means that not only the child born, but his future children, like this. So more germline editing will lead to altering the course of human genes. Gene editing has become more efficient, and if it becomes even more efficient, germline editing can be more frequent, altering the course of human evolution with highly desirable features. Like we have now highly desirable variety of rice, wheat, HYV seeds. So like that, highly desirable human beings. So what has been done in this direction recently? Has anyone been already designed? Yes, very much. In 2018, a Chinese scientist by name Hugh Jiankui from a HIV plus father, HIV positive father and normal mother, he created a test tube baby. But he is not an ordinary test tube baby. In fact, there are two girls called Lulu and Nana, whose gene of CCR5 is edited. CCR5 of these two children edited, which means their future children will have those edited genes. The scientists did not follow the internationally accepted procedure, safeguards. He did it on his own. And his justification is that father has HIV and the children shouldn't have. And in the population of Caucasians, there is a modification to CCR5, and that modification prevents HIV. So he said he manipulated this gene in the embryos before they are put back in the mother. Most scientists don't approve of this. There is so much of condemnation. Number one, first thing is that it may not be a case of informed consent from the parents, fully knowing the consequences. What are the consequences? He manipulated CCR5, but this manipulation doesn't make it like the natural gene. It is a modification which is a very different kind of manipulation, not like the one in others who have resistance to HIV. So manipulation itself is not making it like a natural one. And number two, manipulating this may make them susceptible to influenza, which means unpredictable consequences. Unpredictable consequences. And it is also not clear, before he put these embryonic cells into the mother, it is not clear whether all the cells are edited or only some cells are edited. And if only some cells are edited, then you will have children with diverse cells, unedited cells as well as edited cells. So scientists say that this is a hastily done, and more importantly it is unnecessary, because HIV transmission can be stopped through simpler methods instead of gene editing. And then unpredictable consequences and no informed consent. But then this particular editing may be controversial, but because it shows that the editing is possible and creation of test tube babies is possible after editing, means that in future this is going to happen. So gene edited babies is going to happen in future. Not necessarily for HIV, but there are so many children who have potential for certain disorders. Around 1% of children have potential for disorders because of some wrong gene, single gene defect. So if those things can be altered, then they will have a healthier life. So germline editing can start with handling the cases of lethal diseases. This can start with like that, but then it can go to creation of designer babies, more intelligence, things like that. In fact, some think the reason why CCR5 was edited may not be because of only HIV reason, but also because this is supposed to be playing a role in enhancement of brain capacity. So maybe the scientist is involved in exploring this aspect. So though this is condemned, I think in future gene editing is going to be very normal. First test tube baby was born in 1978, and by now around 5 million test tube babies are born from 1978. So like that, germline editing may be common over years, but will that lead to designer babies is a big leap, because designer babies may involve multiple genes, more genes. So, and it may be more costly, more difficult. But one thing is certain, that editing is going to be there. It may be become as normal as test tube baby. Okay. So germline editing in humans is the most difficult thing, but in other species it is going on. Why? Pigs were edited. Pig editing took place in 2017 by George Church. In fact, George Church sees potential of editing in many areas. For example, he says new species can be created. Species that became extinct, like mammoths, can be created. But why should they be created? It is found that creating them and then their ecosystem may stabilize climate, and it may positively impact climate change. This is one. And it is also found that not only new species can be created, but certain undesirable species can be wiped out. For example, mosquitoes can be wiped out. Mosquitoes can be edited in such a way that they give birth to only males. So within few generations they will be wiped out. And new plants. So species elimination, species creation. So it is proposed that man will be able to impact species in a massive way through creating them, through eliminating some, and he will also design himself towards becoming a more capable, more desirable human being. But all these things were being talked about for many decades, but now one technique that came up only very recently gave more optimism to these kind of possibilities. We'll discuss that technique. a new gene editing tool that is giving so much of hope is called CRISPR-Cas9. CRISPR-Cas9. Cutting a DNA accurately using this technology was discovered only in 2012 by these two scientists, Emmanuel and Jennifer Toadla. 2012, only recently. But this is about cutting. Editing is not simply cutting. After cutting, you have to add the desirable one. That was done by Feng Zhong and George H. These two did editing. There was editing before, but that editing was less elegant, clumsy. For example, see what is wrong with this? This letter is wrong. In the old method, if this has to be changed, then something like Gen X, O, M, ME, and then something else, or some other word here, will be inserted. That is, in the old method, if something is wrong, it can be eliminated, but then many unnecessary things will be added because editing is not very accurate. But in the new method, A can simply be replaced by O. Much more efficient, cheaper, faster. But that doesn't mean it has no limitations, but it is simply, in terms of the previous, it is far better. And how did they find this? The scientists found this from the way bacteria is cutting virus. This system is found in immunity of the bacteria, immunity system of the bacteria. Particularly in certain species equally. This technique refers to E. coli bacteria. How is bacteria handling the virus? It is found in bacteria's genome. First of all, how is a virus handled? When there is a virus, body sends certain things, antibodies, and kills the virus. And if a bacteria cannot kill a virus, then the bacteria is killed. If the bacteria kills virus, what happens is that if it is first time killing the virus, it keeps genome of that virus, a copy of the virus. Say something like this. This is the bacteria. Virus 1 attacked. This bacteria killed virus 1. Then, if bacteria killed virus 1, so next time when virus 1 happens, then the bacteria, without taking much time, can wipe out. And to facilitate that, genetic signature of V1 is stored in the genome of the bacteria. It means if the bacteria has won a battle against virus 1, its genome is stored, its genetic signature is stored. Suppose it wins a battle against virus 2, then virus 2 genome also stored, then virus 3. So bacteria maintains a history of its successful battles. It keeps a record of the kind of viruses it killed in a part of genome. Okay. But before knowing that it is storing genetic signatures of the virus, scientists knew at one part of genome of bacteria, it is something, one thing like this, one thing like this, one thing like this, and then some common thing is 4, something like this. So they found a particular kind of a pattern. It is same thing. Same thing is repeated here. These are repeats. And between the repeats, something else is placed here. These are called spacers. These are called spacers. These are called spacers. So scientists found this structure in 1987. And later scientists found, this repetition they found in 1987, much before it was called CRISPR, and later it was called CRISPR, and also they understood these spacers are nothing but the genetic signatures of the virus. So they found this entire thing is like a vaccination card of the bacteria. The record of its successful battles against the virus. So it is in this context, this particular word is coined. CRISPR is an acronym for clustered, regularly interspaced short palindromic repeats. Clustered, regularly interspaced short palindromic repeats. So these repeats are interspaced. This is being repeated. Repeated means same. In between, these are spaces. So this clustered, regularly interspaced short palindromic repeats. Of course, big name, probably they didn't think at that time it is going to be so frequently used. Otherwise, they should have given a short name. So it was like this. Clustered, regularly interspaced short palindromic repeats. So what may not be clear is what is this palindromic repeat? Palindromic repeat is this consists of palindromic sequences. palindromic sequence has certain biological properties in managing defense here, in managing the immunity. What is a palindromic? Palindromic, in mathematics, it is a palindromic. You read this way, read that this way, it's same. 1, 2, 3, 2, 1 is also palindromic. You can read either way, it is same. That's palindromic. But in genetics, palindromic is different. GATTC. Suppose this is there on one strand of DNA. On its complementary strand, there will be CTTAAG. This is on one strand, its complementary strand is like this. If you read, it will be GAATTC. But if you read this way also GAATTC. This is what is palindromic. It is not read like this, but read on the complementary strand. So if the DNA is like this, it has certain specific advantageous properties. So this is made of palindromic repeats. Certain things are not palindromic. For example, GGCG. It will be CCGC, GGCG, but it is not palindromic. Okay, so not all sequences are palindromic. So this is about clustered regularly interspaced short palindromic repeats. This is how the structure is. This is there at a particular part of bacteria's genome. Okay. But how is all this managed? This is managed by some genes here. These are called CRISPR associated genes, Cas genes. So these Cas genes run this whole system. And also the order of changing the genome also is like this. More recent ones kept near the genes. And Cas9 is one of the genes. The most popular technique is developed with CRISPR-Cas9, but Cas9 is not the only technique. Some, for example, are developing what is called Cas3. And Cas3 can cut in a different way than Cas9. For example, Cas9, it seems, it snipes. It cuts at precise locations. Snipes. How the Cas9 CRISPR system works is like this. This is what is going on naturally. So scientists observed this Cas9 is generating a system. It is like this. Cas9 sends, the system works like this. It has two RNAs. It is a protein with two RNAs and goes to the virus and attaches in such a way that one of this RNA is what is called guide RNA. That is, one of this RNA is what matches with the virus. And there is another RNA. So together they go and attach, this is attached, Cas9 is attached to that part of the virus, matching with this, and simply cuts it off. So virus is destroyed. Remember, bacteria is not doing anything editing. Bacteria is simply destroying the virus. This is how Cas9 works. But Cas3 is supposed to be working differently. For example, Cas3, it seems, it shreds numerous bases. For example, around many thousands of bases. Thousands of bases can be simply shredded by Cas3. So the point here is that there are CRISPR associated genes here, and genes are doing different function. So from the perspective of gene editing, CRISPR-Cas9 is not the last word. CRISPR-Cas9 is popular. It can be CRISPR-Cas3 one day, but later some other gene. In fact, this Cas also is not the last word. And though CRISPR is common, the associated genes will be different in different species of bacteria. Scientists are already trying, for example, using a different species of bacteria with CRISPR CPF1. Okay, so essentially CRISPR Cas or CRISPR other is a way of making use of the system in bacteria, different species of bacteria, and then to cut what the scientist wants to cut. Look at the system here. Naturally, it will have guide RNA from one of these viruses. But suppose scientist makes a synthetic RNA, then Cas system will make guide RNA which is equivalent to synthetic RNA, and then this go on cut. That's what it does. So it means what Cas9 is doing naturally, cutting off the virus, scientists use it to cut the DNA that they want to cut. Okay, so that is about cutting. Cutting will only kill a virus, but if you cut, it just disrupts. But editing is something more than that. If you only cut a particular part of DNA, then what happens is that then they will combine. But when they combine in ways that there can be many undesirable mutations. So scientists want to send another RNA and then place it exactly the way it is cut, exactly at the place it is cut. This includes which means this is editing. So cutting is one, and then replacing, which is editing. Okay, so this is the thing that is developed very recently, and then this CRISPR and CRISPR-Cas system is being used in many life forms, in plants and animals, and for various therapeutic approaches in human beings to produce medicines and things like that. Okay, so this is the one which has raised new hopes about more efficient gene editing. But we'll see if it really creates something like Homo Deus. See, to the question, will genetics create Homo Deus? My answer is no. To me, the answer is clear. Why is it so? See, I am reminded of one slogan by Indian government. It said, "Health for all" by 2008. Health for all by 2008. Did we achieve it by 2020? No. Is it because the government is inefficient, corrupt, that we didn't achieve? I don't think so. It may have been inefficient, corrupt, but this goal itself is defective. Health for all by 2000 AD is a meaningless slogan. Why is it so? What is the problem with this? When you coin something like this, you have particular diseases in mind and you think that those diseases can be cured or vaccination possible by that time with sufficient infrastructure. That can be achieved. But health doesn't work like that, because what contributes to ill health, sickness, itself changes. So new diseases come, new health issues come, environment changes. So environment is not static, and when we change, we create a particular lifestyle, and that particular lifestyle creates health problems like obesity related now. So if you say I will end malnutrition, that is a reasonable goal, but I will end diseases, that is not possible, because if malnutrition ended, then disease can come from a different source. Okay, so to me, Homo Deus concept also is like that. You have some particular genetic diseases, you have some issues. Through genetics, you may change, but then the human body diseases are not going to be constant. Nor environment is going to be constant. So body will have more diseases, different issues, environment will be different issue. So body is changing, and environment is changing, climate is changing. So what we do is a part of intervention, that's all. And the same thing is about how do you intervene in the environment. You wipe out malaria leading to malaria generating mosquitoes, but then when you wipe out these mosquitoes, some other kind of species will come, and we don't know what kind of diseases they will give, but it does not mean we should wipe out. But the issue is that it is a part of struggle. Man has been struggling with the environment, man has been struggling over health issues. So you are not going to have such a system where you will achieve immortality and you will have complete control over environment and you become godlike. It's just not at all like that. So the highest thing that you will know through gene editing will empower you to give more health, to increase probably increase life expectancy, or may contribute to population. So it is not that you acquire godlike features and you have complete control over the environment. It is not all going to be like that. So that saying theoretically. But now look at the present context now. The present context of climate change and globalization, interlinkages, kind of viruses like corona, climate change. So environment is hitting with vengeance. That's how the world works. That's how it is. So it is not going to remain a constant where you keep on improving yourself. You keep on improving yourself, and then there are feedback systems that will give you negative feedback and create undesirable consequences. So it is not that you will create Homo Deus through genetics. Genetics will remain as one aspect of medical technologies and nothing more than that. But does it mean humans are not evolving? No, they may be evolving. But we may find it difficult to see the trend And even if we see the trend, we may find it difficult to evolve in a planned way, because our action is simply part of total system. So we act in a particular way, system acts in another particular way. So as a result, something else is created. So the idea that we control life, we control our evolution, and we are definitely moving in a progressive direction is not at all likely. In fact, given the climate change, it is not necessary we will survive for hundreds or thousands of years. So I think having named ourselves Homo sapiens, sapiens means wisdom, it is enough if we live up to our name instead of trying to become Homo Deus. Thank you.